Hello everybody, I hope you all are doing good. So this is going to be my first video in which I'll be discussing about acute inflammation. I'll be making 4 or 5 of these videos in which we will discuss literally everything you need to know about acute inflammation. So I would like to begin with a question towards you. What is inflammation? What is the definition of inflammation? Because we know what is inflammation, but sometimes we cannot define it in a right way. We cannot define it properly. So let's just learn what is the definition of inflammation. Inflammation is a response of vascularized tissues. So it is a response of vascularized tissues to what? To infections and damaged tissues. Or you can say to offending agent, in which you bring cells and molecules of host defense from the circulation to the sites where they are needed, or to the sites where offending agent is present, in order to eliminate the offending agent. Let me explain this to you really nicely. Okay. I said that this is the response of tissue in which you have offending agent, okay, whether it is infectious agent or whether it is injury, okay. Suppose this is your liver, okay, and the liver has some kind of offending agent, whether you suppose it as like infectious agent or maybe these are the injuries that the liver has suffered. So the liver has offending agent, okay. Now what the hepatocytes will do, hepatocytes will secrete some kind of chemicals which will bring the components of host immune system, components of host defense, which are your leukocytes, which are your WBCs. Suppose this is your vessel wall, this is your blood vessel, and within the blood vessel you have your leukocytes or WBCs. Okay, liver will try to bring these leukocytes out of the vessel wall towards itself because it has the offending agent. So this is inflammation. This is it. Inflammation is simply a process in which you bring leukocytes or you bring the components of host immune defense towards the site of offending agent. Okay, you literally brought these leukocytes towards the liver, which has the offending agent. Now why do you want to bring these leukocytes towards the liver? Why? Because you want leukocytes to engulf these offending agents. Okay, you want phagocytes to eliminate these offending agents. This is the simple definition of inflammation. Okay, let's read it again together. What is inflammation? It is a response of vascularized tissues to infections and damaged tissues that bring cells and molecules of host defense, which are your WBCs, from the circulation, because WBCs are present within your blood vessel, within the circulation. So you will try to bring WBCs from the circulation to the sites where they are needed. Why? In order to eliminate the offending agent. Okay, I hope this is pretty clear. Now, we think inflammation is a destructive process, but in actuality, inflammation is a protective mechanism, because with inflammation, you clear the infectious agent, you clear the injured cells, you clear the necrotic cells. Okay, so this is a protective mechanism, but obviously it has some local tissue damage as well. Okay, look at this diagram again. Okay, what are the causes of inflammation? To understand the causes of inflammation, you must have a good concept of antigen. What is an antigen? Antigen is any substance which is able to elicit an inflammatory or immune response. Okay, now antigen can be exogenous, it can be endogenous. Exogenous antigens are bacteria, virus, parasite, fungus, allergens, foreign body. And endogenous antigens are necrotic debris. Necrosis can occur due to any cause like ischemia, injury, trauma, burns, irradiation. And in the cases of autoimmunity, obviously the antigen is endogenous. In autoimmune reactions, your WBCs are reacting against your own cells, okay, your own organs. So in autoimmunity, the antigen is endogenous. Okay, what are the components of inflammation? Okay, we all know that inflammation is not always going in our body. To begin inflammation, you need to bring some changes in your body so that you can start inflammation. Okay, so first thing in which you want to bring changes is your blood vessel. And then your leukocytes. Let me explain this to you really nicely. Okay. We all know that leukocytes are present within our vessel, right? We all know that, within our vessel. And vessel wall is tight compartment. The vessel wall is not letting the WBCs go out of it in normal circumstances. So to begin inflammation, you will want to make this vessel wall permeable. Okay, suppose that in normal circumstances, this vessel wall is like this, very tightly adherent. The cells are very tightly adherent to each other. And during inflammation, you will make it somewhat like this. You will want to create some gaps between the cells so the leukocytes can easily escape out of the vessel. From here, okay? This is the first change our body makes. I will explain how this change is made. I will explain everything. But just remember, the components of inflammation mainly are two: your blood vessels and your leukocytes. Okay? In blood vessels, you will like to make it more permeable, so it will be very easy for the leukocytes to escape, because we want them to go at the site of offending agent. Okay, leukocytes are just escaping out of these spaces. First thing is this, okay? And under normal circumstances, your leukocytes are present in the central portion of bloodstream. Okay, they are not in contact with your vessel wall. In order for them to escape out of vessel wall, they should be here at the margins of the vessel wall. Okay. Next thing you will do is you will bring these leukocytes towards the margins. Okay. Is that clear? First thing, you made this vessel wall permeable. Second thing, you try to bring these leukocytes towards the margins. Okay, towards the margins. But how do you bring them towards the margin? It's very easy, because you will, okay, let me just clear some of it. Let me just clear a whole of it actually. Okay. When you create these gaps in the vessel wall, when you create these gaps, suppose leukocytes are in the central stream. When you create these gaps, not only leukocytes can escape. First, before the leukocytes, fluid will escape of here. All the fluid that is present over here will escape. And eventually leukocytes will come at the margins. Okay, this is the one way of bringing leukocytes towards the margins. And the second way of bringing the leukocytes towards the margin is simply adhere them to these cells. But how do you adhere them to these cells? Okay, inflammatory cells, or you can say the cells which are suffering damage or which are having the offending agent, secrete some kind of substances. They are secreting some kind of substances. What do these substances do? These increase some molecules on the leukocytes as well as on the endothelial cells. Okay, as well as on the endothelial cells. These two bind together, and eventually leukocytes are towards the margins of the vessel. This is what we need, right? This is what we needed in the first place. We want to bring these leukocytes towards the margins. Is that clear? So the changes in the vessel wall are, you want to make it more permeable, and changes in the leukocytes are you want to increase some adhesion molecules on the surface of leukocytes so that we can bring them towards the margins. I hope this is pretty clear now. Okay, if something is not clear, you may ask in the chat box, you may ask in the comment section. Okay. Okay. So we discussed blood vessels and we discussed leukocytes. So what are the leukocytes that are present in a body? And what are their roles? Okay, there are so many leukocytes like neutrophils. Neutrophils have role in acute inflammation and they have role in bacterial infections. Okay, macrophages have role in chronic inflammation. Lymphocytes have role in viral infections, hypersensitivity reactions, and chronic inflammation. Mast cells and eosinophils have role in allergic reactions, that is type 1 hypersensitivity reactions. And obviously there are other components of inflammation as well, like dendritic cells, natural killer cells, complement cascade, cytokines and chemokines. Just look at this image again and try to recall everything we discussed just a moment before. That's great. Let's just move forward. Okay, so my question is, you know what inflammation is, right? You know that how we begin inflammation, what are the components of inflammation, in which component you want to bring which change, you know everything, right? But my question is, how do our body cells recognize any antigen? How? How do we know that this is an offending agent? How? Okay. Antigen can be extracellular, antigen can be intracellular, right? Suppose this is your cell, and outside the cell you can have extracellular antigen, and within the cell you can have intracellular antigen, right? So to recognize these antigens, your cells have special kind of receptors. To recognize extracellular antigen, you must have receptors on the plasma membrane, okay, projecting into the interstitial space, like this. Okay, and to recognize intracellular antigens, you must have some receptors present intracellularly. I hope this is clear. Okay, so do you know any receptor that has role in recognizing microbial products? Very important is your toll-like receptor. Toll-like receptor has the ability to recognize microbial products. What kind of microbial products? Like lipopolysaccharides, peptidoglycan chains, okay? This has the ability to recognize products related to microbial, related to microorganisms, okay? Then you have nod-like receptors. Nod-like receptors can be intracellular. When they are intracellular, they recognize some intercellular organisms like Listeria. They recognize the products related to Listeria. And when they are extracellular, they simply recognize the peptidoglycans of bacteria. Okay. Then again you have one more, Rig-like receptors. Rig-like receptors recognize nucleic acid. They recognize nucleic acid, RNA or DNA. So that's why they have role in recognizing viral antigens, or they elicit the antiviral response. Okay, is that clear? One more thing that I would like to tell you is Rig-like receptors, when they are stimulated, means when they capture an antigen on whatever cell surface they are present, that particular cell starts secreting interferons. Okay, interferons. So, and interferons have the role in generating antiviral response. Okay, these are all the receptors related to related to microorganisms. Okay, These are related to microorganisms. But in the definition, we learned that inflammation is not only required in infections, but inflammation is also required in necrosis, in injury. So what are the components that will elicit inflammation in cases of injury? Right? This is a very important thing. Let me just clear all of this. Okay. You know that during injuries, the cell that has suffered injury will secrete all of the substances which are normally present in the other cells as well, right? Like potassium that is present normally within the cells, or like ATP that is also present in normal cells, right? And uric acid. Okay, let me explain everything to you clearly. You know that DNA is normally captured within the cell. Okay, your ATP is captured mostly in your cells, and your potassium is also in your cells. So whenever you find uric acid out of your cell in the interstitial compartment, it is an indicator of DNA breakdown. You know that uric acid is formed whenever there is a breakdown of DNA. So this indicates that the cells, that the neighboring cells has suffered some kind of injury. Okay, and if you have ATP, a huge amount of ATP is present in interstitial space, it indicates that mitochondria has been damaged, because we know that mitochondria has a huge amount of ATP within itself. And if you have less concentration of potassium within the cell, that cell is indicating that I have suffered some kind of plasma membrane injury. Is that clear to you guys? So our body has some special receptors to recognize these things. Whenever our body recognizes these things with a receptor, an inflammatory response is generated. Okay, so I think this is pretty clear now. Let's just read the text together. How do cells recognize an antigen? This is my question. There are some special kind of receptors. If the receptors are present on plasma membrane, they are going to recognize extracellular antigen. And if they are present within the cell, in the endosome, they are going to recognize intracellular antigen. Okay? For microbes, the most important one is toll-like receptors. Okay? Then you have another kind of receptors known as knot-like receptors. They recognize the peptidoglycan of bacteria. They also recognize intracellular microbes like Listeria. Then you have rig-like receptors, which recognize microbial nucleic acid, and they have important antiviral response. I told you one more thing, that whenever this receptor is stimulated, the cell onto which this receptor is present, that cell will start secreting interferons, okay, like interferon alpha and interferon beta. Interferons have very important role. Particularly these interferons have very important role in generating antiviral response. Okay, then we talked about then we said that these are only microbial antigens for which we have receptors. Like no, actually this. Then I said, what happens if we have necrotic debris? How do our body cells recognize necrosis? What are the indicators of cell damage? Sorry, we discussed that whenever there is high amount of ATP present, this indicates that there is the damage to mitochondria. If we have uric acid, that means DNA damage has occurred. If we have reduced potassium ion concentration within the cell, that means there is damage to plasma membrane. Such cells are recognized by special kind of receptors. Okay, next point is very important, like very, very important, and we need to understand it very nicely. Let me clear all of this. Okay. You know that if someone has suffered any kind of infection, their body will make antibodies, right? I'm not talking about how does our body make antibodies. I'm just talking about that we have made antibody once we have suffered some kind of infection, right? So what is the function of antibody? Antibody structure is somewhat like this. This is the Fc portion of antibody, okay? This is the Fc portion. Antibody itself doesn't have the ability to kill the microbial product or any kind of organism. Antibody doesn't have that ability. Antibody is just there to capture the offending agent. Now just suppose someone suffered mumps virus infection. Okay, whenever someone suffers mumps virus infection, they are able to make lifelong immunity because they make certain kind of antibodies against mumps virus. Okay, now if next time, after some time, the mumps virus again comes into the body, what will happen? These antibody will capture the mumps virus. Okay, antibodies do not have the ability to kill it. Okay, just remember this: antibodies are only working as opsonin. What is opsonin? Opsonin are the substances which make the phagocytosis of certain substances easy. Okay, suppose this is your phagocyte, neutrophils or macrophages. Neutrophils or macrophages, these are your this is your phagocyte actually, not phagocytes. This is your phagocyte. Okay, now phagocyte has some important receptor for this Fc portion of the antibody. Now suppose someone has the mumps virus again and the antibody captured this mumps virus. Now antibody will bind to this receptor on the phagocyte with its Fc portion, and suppose this is, and then what happens is your phagocyte will capture all of this thing, will engulf all of this thing and destroy it out. This is the role of antibody. So you must have the receptor for Fc portion of antibody. Otherwise, what will be the benefit of making antibody? Because antibody is not able to kill organisms by itself. Rather, it presents those, rather it binds to the phagocytes. It captures those organisms and binds to the phagocyte. And then phagocyte will do rest of the job. It will engulf it and destroy it. I hope this is clear.